everyone, welcome back to my channel. In this video lecture, we will cover a new chapter of biology, that is biotechnology, principles and processes. So, there are two principles of biotechnology: genetic engineering and maintenance of sterile environment in chemical engineering processes. The basic principles of biotech. Genetic engineering allows the isolation and introduction of only the desired gene into the organism without introducing the undesired genes. Steps involved in genetic engineering are development of rDNA, cloning of desired gene, transfer of gene into suitable host organism. Then origin of replication, ORI, is a specific DNA sequence in a chromosome that can initiate DNA replication. Then cloning, the process of making multiple identical copies of template DNA. Plasmid, a circular extra chromosomal material that is capable of autonomous replication. They are used as vectors for cloning and expression. Antibiotic resistance genes, the gene in certain microorganisms that bestows on them the ability to grow in the presence of specific antibiotics as the gene gives them resistance. Then restriction enzymes. They are the enzymes that cut DNA at specific fragments. They are also called as molecular scissors. Vector plasmids that are used to multiply and transfer the desired gene from one organism to the other. Then ligase, enzyme that are responsible for joining of the desired gene fragment with the host DNA. Then the basic step in genetic modification includes identification of desired DNA fragment, then introduction of desired DNA fragment into suitable host, then maintaining foreign DNA in the host and transfer into the progeny. Then endonuclease cut the DNA in the middle, where exonuclease cut at ends. For example, E. coli 1, Hind 3. Restriction enzymes cut at a specific point, specific site on DNA known as restriction site. Ligase is the enzyme that joins two DNA fragments. Then agarose gel electrophoresis in DNA. Then cloning vector. Vector is the DNA molecule, carries a gene of interest. For example, plasmid. Plasmid is autonomously replicating extra chromosomal genetic material. It is different from chromosomal data, used as a vehicle for transfer of gene of interest into the host cell, contains ORI, site where replication begins, and it also contains antibiotic resistance genes. Then selectable markers, they are used to identify transformed cell. Transformation is the process used to introduce piece of DNA into the host cell. The gene encoding resistance to antibiotics such as ampicillin, chloramphenicol are some of the useful selectable markers. Then vectors for cloning in plants. Agrobacterium tumefaciens is a pathogen of several dicot plants. It is used as vector for the plants, and it can deliver a piece of DNA known as tDNA, that is transfer DNA, tumor DNA, to transform normal plant cells into tumor and direct these tumor cells to produce the chemicals required by the pathogen. Then the competent host. In order to allow bacteria cells to take up the DNA, bacterial cells should be made competent. Then this can be treating the cells with specific concentration of divalent ions such as calcium, which create pores in the cell wall in the bacteria. Then such bacteria are subjected to heat shock. Then calcium treated competent cells are kept in ice. Then they are incubated at 42 degrees Celsius for 1 to 2 minutes and then immediately placed on ice. This forces the rDNA into the competent cell. Apart from this, DNA can be inserted into host cell using ballistic guns, microinjection, gene guns, etc. Then process of recombinant DNA technology. First, several steps included. First, isolation of genetic material. To isolate DNA, membranes need to be broken down. Cells can be treated with lysosomes, cellulose and chitinase. Then ribonucleases are used to remove RNA, where proteases are used to remove protein. After this, pure DNA obtained through precipitation through ethanol. Then DNA is then obtained as fine thread in suspension, like this. Then second step takes place, that is the restriction digestion of isolated DNA. Agarose gel electrophoresis is used to check the progression of restriction enzyme. Then the gene of interest is now inserted into the specific vector. Then it is joined through an enzyme known as ligase. Then amplification of DNA of interest takes place using PCR. PCR is the polymerase chain reaction used to amplify the target gene of interest. For this, two set of primer, forward and reverse primers are used. Then DNA polymerase enzyme is used to amplify the DNA. And the most common polymerase used during PCR is Taq polymerase. Then insertion of recombinant DNA is into the host cell organism. Then the recipient cell is made competent to take up the recombinant DNA. The expression of desired protein is the fifth step, is the ultimate goal of rDNA technology is used to obtain desired protein of interest, and the protein obtained is known as R protein. Then to produce large component of recombinant protein, large vessels are formed, that is known as bioreactors. Bioreactors provide optimal conditions for achieving the desired product by providing optimal growth condition, that is temperature, pH, substrate, salt, vitamins and oxygen. Basic parts of bioreactor are agitator, oxygen control system, foam control system, temperature control system, pH control system, sampling pot, inlet and outlet. Bioreactors are of two types, stirred and sparger. Stirring type are the stirrer is fixed to a bioreactor having a curved base to facilitate better mixing. It also improves aeration of the medium. Then sparge type of bioreactor. In this, air is bubbled into bioreactor from the base, and the bubbling of air results in mixing as well as aeration. Then the downstreaming process, the process and method involved in the separation and purification of the desired products are called as downstream processing. In case of drugs, the products needs to be suitably formulated and drugs tested before being made available commercially. So this much for today's video. Hope you liked it and understood it. Thank you for watching my video.